Of the week is upon us here. Good morning, everyone. Happy New Year. So we're going to have a fantastic year in the coming year. So whoever celebrates, we're with you guys today. And we went to many different places, different countries, different speakers, different branch of medical sciences. But I have to say that one of the best pharmacologists in the country is right here on the third floor, Daniele Piomelli, and we're so delighted that he agreed to tell us about his research and in general about the pharmacology that is of course of interest to all vision people. And so with that, I would like to pass this to Zach. Zach.
Good morning, everyone, and welcome to this week's ophthalmology seminar. It is my pleasure to introduce one of our own UCI professors, Dr. Daniele Piomelli, as our speaker. Dr. Piomelli initially completed a PharmD at the University of Naples, and then went on to obtain his PhD in pharmacology at Columbia University, where he worked in the lab of James Schwartz. In the Schwartz lab, he focused on characterizing the effects of 12 lipoxygenase pathway products on the transduction of histamine responses in the marine mollusk Aplysia. He then went on to work as a postdoctoral researcher in the lab of Paul Greengard at Rockefeller University, where he worked further on lipoxygenase metabolites in neuronal transmembrane signaling and in the bidirectional modulation of phospholipase A2. He continued his distinguished career in research at INSERM in Paris, and then at the Neurosciences Institute in La Jolla, and has since made important biochemical discoveries regarding the biosynthesis, metabolism, and pharmacological relevance of neurotransmitters such as anandamide. Additionally, he has pursued many cannabis-derived compounds as modulators of neurological function. Dr. Piomelli has accrued over 45,000 citations, has an H-index of 99, and 34 patents. Additionally, he has served as co-founder for several pharmaceutical companies and is currently serving as editor-in-chief of the journal Cannabinoid Research and as the director of UCI's Center for the Study of Cannabis. Much of his research demonstrates the diverse neuromodulatory effects of endogenous cannabinoids, and I'm excited to see how these molecules can be repurposed for ophthalmological applications. Without further ado, please welcome Dr. Piomelli.
Hello, everyone. Thank you so much for having me here. May I share the screen now? It looks like I am allowed to do that now. All right. So again, thank you very much for having me here for this seminar in your center. Thank you, Chris and Zach, for the nice and thorough introduction. I was asked by Chris to give to this distinguished audience an intro or a primer on cannabis, which I'll be very happy to do. I also asked whether it would be okay to present some of the work we have done, and because in an hour or 45 minutes I cannot do both of them, both tasks, I picked one area that I think might be of interest to all of you, but of course, it's just one area, so we will, if you have questions on other areas, I'll be very happy to entertain them. So let me start off by looking at the history of cannabis, which is always a good place to start. And I will give you in this slide a short history of the last couple of centuries of the interactions of humans with cannabis. We have used as a species cannabis for the last 11,000 years for a variety of reasons. Cannabis is a source of food. The seeds are very nutritious. It's a source of fibers. The stem can be converted into textiles that have been used for thousands of years. And of course, it's also a source of psychoactive compounds and medical compounds. But cannabis became known to European science, which at the time in the 19th century was basically all the old science that there was, in the mid of the 19th century, about 1845, when a French author wrote a lovely book that described the physiological properties, mostly the neuropsychological effects of cannabis in great detail. The name of that author is Jacques Moreau de Tours, and the title of the book is Hashish and Mental Disease, or Du hashish de l'aliénation mentale. So that along with other texts produced in the same time, introduced cannabis to European science, and was the beginning of a series of investigations into its pharmacological properties proper, discovery of its analgesic effects and spasmodic effects that led to the introduction of cannabis in the United States pharmacopoeia in the middle of the 19th century. 1854 is the first year where the US pharmacopoeia introduced it. Mostly as an analgesic and antispasmodic medication, where it stayed until 1942. So the reason why it was removed from the pharmacopoeia is complex, but the main reason is really not scientific. It's not a pharmacological reason, not because, for example, it was found to be toxic, but was really the consequence of a political shift in the United States and in the rest of the world. The introduction in the US of the so-called Marijuana Tax Act of 1937 made cannabis illegal and basically stopped most research on cannabis. Most, but not all research. And luckily, some investigators, for a variety of fortunate circumstances, were able to continue working on cannabis. And between 1944 and 1964, finally, the active component, the active constituent of cannabis, that is the compound that is responsible for the intoxication induced by cannabis, but also by most of its other pharmacological effects, was discovered. And that compound is delta-9 tetrahydrocannabinol, or THC for short. And two main individuals are involved in that discovery. Roger Adams here in the United States, who described the chemical structure of THC in about 90% accuracy. And then the last 10% was Rafi Meshulam in Israel, who identified a critical double bond that is present in the structure. I'll show you the structure in a minute. You will see what I'm talking about. But the discovery of THC happened despite this very unfriendly political landscape, which was confirmed, a landscape that was confirmed in 1970 under the Nixon administration in the so-called Controlled Substance Act. The reason why this act was enacted is because actually the Marijuana Tax Act of 37 turned out to be unconstitutional for a variety of reasons. So the Nixon administration had to find a new legal way of stopping cannabis use in the population, and that was the CSA of 1970. This really slowed down research, and in fact, we still have a research lag that is largely due to this very restrictive legal control. And by very restrictive, I mean very restrictive, because as you probably know, the CSA established schedules from 1 to 5, and the top one, number 1, Schedule 1, is the most restrictive one and includes substances like LSD, MDMA or ecstasy, heroin, and cannabis. Now, for reasons that I hope you will see articulated later on in my presentation, that is not a scientifically based scheduling, but of course that's the rule of the land, and that's the rule we have. And it caused very substantial delays in scientific progress. Again, delays. At the end of the day, scientists are a resilient bunch. We really are hard-headed in many ways. And so between 1988 and 1990, mostly through the work of Lynn Howlett here in the United States, cannabinoid receptors were discovered. Eventually, they were cloned by Lisa Matsuda and others. And that was really important because, of course, that demonstrated that the mechanism of action of THC was mediated by a receptor, just like the mechanism of action of other plant-derived compounds like morphine or cocaine or nicotine, et cetera, et cetera. So they put cannabis in sort of in a well-established tradition. And that well-established tradition led to investigators to ask, is there an endogenous system that activates those cannabinoid receptors? And between 92 and 1990, again, a series of accumulated discoveries led to the identification of this brain and peripheral signaling system, to which I will give you some details later on. But you probably have appreciated from this timeline that we would be in a different place if it had not been so hard to work on cannabis. And just want to make very, very clear, the legal landscape is not really an obstacle for people getting cannabis, because you probably know that it's quite easy to buy and get cannabis. Was easy before legalization in most states of the union, and still easy now. It's still easy. It's even easier now. But really what it did, it was to impede research, and that's really a problem. So now we are in 2021 with 48 states of the union having legalized cannabis for medical uses in various forms, either the whole plant or hemp or components of the plant like cannabidiol. And we still don't have fundamental answers to important questions and related to the health effects of cannabis. But we do know a whole lot. We do know a whole lot despite these delays, and we know how cannabis works. And by that, I mean, we know how cannabis produces the effects for which is mostly used, which is an intoxicating action. And what we know is that this molecule you see here on my screen, on the screen, is responsible for those effects. This is tetrahydrocannabinol, or THC. And by the way, this is the double bond that escaped Roger Adams and was eventually identified by Rafi Meshulam. So if you look at the structure of THC, those of you who are chemically inclined, and many of you may, recognize right away that it is actually quite different from most other plant-derived phytochemicals that have medicinal use. I'm thinking about morphine, nicotine, cocaine, various other alkaloids that are derived from plants. THC is not an alkaloid. You see there is not a single nitrogen molecule here, a nitrogen atom here. The molecule is instead a terpenophenolic molecule compound, so it's a benzopyrene derivative. And the terpenophenolic molecule means that it's essentially lipid in nature. It's a lipidic molecule, and that really had an impact on its discovery, made it hard to discover, but also it made it hard to understand. And we will see to a certain extent how that was a problem. We know now that THC binds to cannabinoid receptors. I mentioned them before, and what you see here is actually an imaging of a PET imaging of cannabinoid receptors in the human brain. And I don't know if you can appreciate this, but this is a classical pseudo color scale with, red and yellow being higher densities. You see that the receptor is quite abundant throughout the cortical mantle, is also present in the cerebellum, but it's actually kind of remarkably sparse in the pons and in the brainstem. And that is also interesting for a variety of reasons, which I will point out to you. So this is how cannabis works. THC activates cannabinoid receptors. So let's take a look a little bit closer to cannabinoid receptors. The receptor I just showed you, this receptor here, is now called Cb1 receptor, was the first to be identified and cloned. But a few years later, that happened actually in 1990, and 3 years later in 1993, Cb2 receptors were identified, and they only share about 40% sequence homology with Cb1. They're very different. And what you see here, of course, these are not real X-ray structures for these receptors because they have not been fully, actually Cb1 has been the structure has been fully resolved, but not Cb2. But they differ. These are models, but to show you how different they are, they're still 7 transmembrane domains spanning receptors. They're coupled to G proteins, GI, GOs, and they also have a full repertoire of intracellular transduction mechanisms. They differ quite a bit in their distribution. Cb1 receptors are present pretty much throughout the whole body, but they're very abundant in the brain. They're also expressed in peripheral neurons, but in cells that you would not expect to see, like adipocytes, for example. These are the cells in white adipose tissue, hepatocytes, the cells in our liver, and also other cells. But it turns out actually that in those cell types, Cb1 receptors exert really important physiological functions. Cb2 is much less ubiquitous in its cellular distribution because it's predominantly expressed in macrophages / monocytes and B lymphocytes. So in cells of the innate and adaptive immune system. I don't want you to get the impression that then is less, Cb2 is less abundant or less, it's less abundant for sure, but it's not less distributed than Cb1 because macrophages are present everywhere. So when THC floods the body, it activates Cb1 receptors and Cb2 receptors throughout the body. And that's something that has interesting and potentially risky repercussions in terms of health. So what do Cb1 receptors do? I will just give you a nugget of information that distills many, many years of research on this receptor. It is involved in the regulation of stress in unmistakable ways. It's really very important in stress control. It's also involved in pain control, and I will say a few more things about that later on, but it's also very important in energy balance. And in fact, the first drug ever to target cannabinoid receptor, a Cb1 receptor antagonist called rimonabant, was indeed quite effective in reducing obesity, but had also side effects due to its penetration in the brain and the blockade of the stress coping effects of Cb1s in the brain. So in the brain, Cb1 receptors are very important in modulating stress response. If you block them, you end up with a lot of situations like anxiety, depression, irritability, suicidal ideation. But if you block it outside the brain, mostly in the adipose tissue, you can control adiposity. So it's a kind of an interesting but complex situation. And for Cb2 receptors, the main function is immune response, modulation of the immune response, immunomodulatory effect, as well as an analgesic effect. So this is another view of the Cb1 cannabinoid receptor in the brain. You have seen the PET scan before. This is a scheme. The point here is to show that the receptor is present basically throughout the brain, but also is not present in certain specific regions. You see here in the pons and the medulla, the receptor is less expressed. And in particular, I'd like to point out this particular nucleus, the Prebetzinger complex, which regulates the respiratory rhythm. And there are tons of opioid receptors there. And that's one reason why opioid overdose almost inevitably is lethal, because it causes respiratory depression and respiratory blockade. But there are no cannabinoid receptors in the Prebetzinger complex. And that's the reason why it's very hard to kill people with cannabis. Killing yourself with cannabis is not a very effective strategy. And although cannabis can be toxic chronically, it has very little lethality at all. So, and that's something that we should also keep in mind. So at a very broad level, how do brain cannabinoid receptors, Cb1 receptors, work? So what you see here is an anatomically faithful localization of the Cb1 receptor. Cb1 receptors are localized to a specific part of the presynaptic bouton, sort of the perisynaptic annulus, the little ringlet that sits around the active zone here. And this receptor controls through either intracellular messengers or through beta gamma, the activity of calcium channels and potassium channels. So calcium channels are closed, potassium channels are open. The net result is a decrease in calcium entry and an increase in potassium entry. You know physiology, you know that that causes a decrease usually in neurotransmitter release, because less calcium, less transmitter release, but also more potassium, more hyperpolarization, less transmitter release. So that is the presynaptic regulation of transmitter release is a pervasive effect of Cb1 receptors throughout the brain. There are very few exceptions. There are few cannabinoid receptors, Cb1 receptors in some specific presynaptic compartments that actually potentiate the release of transmitter, particularly glutamate. It's an exception. And there are few cannabinoid receptors that are not presynaptic, they're somatic, but their role also is exceptional. So if you want to bring a message home that is there to stay, is that Cb1 receptors are presynaptic regulators of neurotransmitter release, regardless of the neuron type. So it could be glutamatergic, and then you're inhibiting excitation. It could be GABAergic, and you're inhibiting inhibition. So it's a mixed bag of effects, but it could also be serotonergic receptors. It could be dopaminergic receptors, dopaminergic neurons, serotonergic neurons. And in that case, you have different changes in the activity of modulatory neurotransmitters. Now, before I move on, I'd like to just make sure that you appreciate that cannabis is a little machine. It's a little factory, chemical factory, that produces a whole lot of so-called secondary metabolites. These are metabolites that are not involved directly in the life of the cannabis plant, but they're important in some other function, which we mostly don't know. And they all share the cannabinoid nucleus you have seen before, this benzopyrene or terpenophenolic nucleus I showed you before. Is shared by about 140 cannabinoid compounds in the cannabis leaf, the cannabis flowers, etc. The cannabis flower is really where most of the activity, the chemical activity is. So some of these compounds are present in high levels, others are present in very, very low levels, but one guy only is THC, is the activator of cannabinoid receptors, and that's tetrahydrocannabinol. Cannabinol, which is a derivative of THC, is also a weak activator, but really THC is the principal cannabinoid receptor agonist in the plant. The other compounds are called cannabinoids, but they're not really ligand of the cannabinoid receptor. And I'm sorry about the confusion, but I didn't make this terminology. I always thought that was a little bit confusing myself. Now, you probably have heard of another compound made by the cannabis plant called cannabidiol or CBD. And now cannabidiol, you can find it anywhere. You can find it in grocery stores. You can find it in your gym. And it's sold for a number of different ailments. Most of that are completely unsubstantiated. They're not grounded on any research, because even though CBD we know has several pharmacological effects, the research really on CBD is nascent. Something that for years was ignored. So a lot of the claims that are made out there are plain wrong or unsubstantiated. So please don't buy CBD for whatever reason, unless you know for a fact that it's effective. And there are only like 1 or 2 conditions where it's effective. And one condition where it is effective, which has been proven by placebo-controlled studies, is the anti-epileptic effect it has in some forms of childhood epilepsy, such as Dravet syndrome. Now, these effects are not mediated by cannabinoid receptors. The effects of CBD, although CBD can modulate Cb1 receptors allosterically, is really not a Cb1 receptor activator. So most of its effects are mediated independently, and a whole host of receptors have been suggested as the effector for CBD. And when you have so many, it means you probably don't have any. We don't really know for sure how CBD works. So the other 138 cannabinoids, I told you about 140, so minus CBD and THC, 138, have effects where we don't really know much about them. And probably it's okay, because it's not that we need to know every single effect of every single molecule that a plant makes. But the fact that this plant makes all these compounds is interesting, and it's attracted a lot of industry attention, as you may probably know. So the fact that we have, back to THC rather, the fact that, and here is our friend THC, now you're familiar with the molecule, it binds to cannabinoid receptors. So why do we have cannabinoid receptors? I told you already that we have cannabinoid receptors because we have an endogenous signaling system that these receptors recognize as ligands, and these are called endocannabinoids. And they are involved now, we know, after about 25 years of work in this, they're involved in a series of quite important physiological functions: feeding, emotion, pain, cognition, rewarding mechanisms. And today I'll be focusing on pain, but these are all very important areas. The fact that I pick pain doesn't mean that the others are not important. So I don't know how many of you are really into hardcore biochemistry, but I just want to give you a sense of what are these endocannabinoids. So there are 2 of them. One, I think they're called sometimes the 2 best known, and there might be others, but they're in small quantities and they're probably less important. But one of them is called anandamide. And anandamide is the first one that was discovered back in December 92. Is an interesting molecule because, as you can see from its chemical structure, it is a lipid molecule, a lipid-derived molecule. It contains arachidonic acid, which is these fatty acids with 20 carbon atoms and 4 double bonds. I am pointing to the double bonds right now. There is an amide bond that links arachidonic acid to ethanolamine. So this compound is chemically arachidinoyl ethanolamide. And the name anandamide was picked by the person who actually isolated it in Bill Devane in Rafi Meshulam's lab back in the 1990s. And ananda comes from a Sanskrit word. Anandamide comes from a Sanskrit word ananda, which means bliss. And I thought it was actually a very fortunate naming, because of what we know of the role of this compound in relieving anxiety, relieving depression, improving mood, and increasing social interactions. So he's actually a good guy, and the name anandamide fits it very well. Now, a couple of years after the discovery of anandamide, the Rafi Meshulam and Sugiura in Japan identified this other ligand called 2-arachidonoglycerol. And I apologize, I flattened the structure of 2-AG here for reasons of space, but this is arachidonic acid, just like this guy here. It's just, I just flattened it. And you see that there is, in this case, arachidonic acid is bound not via an amide bond, but rather via an ester bond to glycerol. So this is 2-arachidonoglycerol, and we didn't find a better name for that, so it's called 2-AG for short. Both anandamide and 2-AG bind cannabinoid receptors. Anandamide prefers Cb1, 2-AG recognizes equally well Cb1 and Cb2. And here are the pathways by which they're produced, and I won't go into the chemical detail, but I want you to appreciate one common theme here. So for both anandamide and 2-AG, there are pre-formed lipid precursors. They're sitting in the membrane. And these lipid precursors are then cleaved upon demand when stimuli arrive. For example, a transmitter receptor is activated, or calcium enters the cell. These enzymes release the compound upon demand. And then the compound, after it's released, activates its receptors. And of course, like every other self-respected transmitter, it has to be cleaved, it has to be deactivated. And for both anandamide and 2-AG, there are two main mechanisms of deactivation: uptake into cells. Don't forget, they're outside, they need to be taken up. They're taken up by cells, and this is not shown in the slide. And then they're cleaved inside cells by enzymes. And in particular, in the case of anandamide, the main deactivating intracellular enzyme is called fatty acid amide hydrolase, and it forms ethanolamine and arachidonic acid. In the case of 2-AG, is an enzyme called monosylglycerol lipase, and it forms arachidonic acid and glycerol. So this is how this pathway is terminated. There are a few other biological mechanisms that through which the compound can be transformed, but I will not go to that today. What I like to do instead is to give you, I know many of you are clinically active. I like to give you a sense of some of the issues, the risks, and the benefits of cannabis before I go and talk briefly about the endocannabinoid, the potential use of the cannabinoid system as a source for therapeutic drugs, particularly in the area of pain. So what I'm pointing out is a report, a pretty hefty report. Here, let me show you the original. Here, this is the thick report published by the National Academy of Science in 2017. Now, 3 years later, is still pretty updated. Unfortunately, I wish that we had a lot more new data. There are a few new data, but not as many as we would like. But this was the product of extensive work by a 16-member panel assisted by NASM staff. And this panel looked at 24,000 primary studies from 2016, focusing on systematic reviews, and issued this 468-page report with 15 conclusions and 4 recommendations. So the areas covered in health effects covered are listed here: cancer, cardiac, respiratory disease, immunity. So, and I urge you to, if you have any interest in any specific areas, to go online. You can get the full report if you go to the National Academy website and look it up, because that is that evidence is vetted. And so instead of running the risk of finding some unsubstantiated claim. If you go there, I think most of that evidence is really believable. But I'd like to just mention one thing to you today, and then it's mental health. And the reason why I want to point that out is because, of course, that's one of the main things that people are worried about. Does cannabis affect mental health? So I'll start off with the bad news. Cannabis does seem to pose a risk for schizophrenia. So especially high use of cannabis during young age, adolescence, and early adulthood is clearly associated, and there is now substantial evidence for that. I would say most conclusive evidence that is associated with the development of schizophrenia. Now, this said, this is not a simple thing. It's not you smoke pot and you go crazy. That's absolutely not the case. But substantial use during critical periods of life, particularly adolescence and young age, when schizophrenia first emerges, that is linked to an increased risk. The risk size varies, but it is substantial. Now, to make things more complicated, however, there is an evidence that is moderate, but it's better than weak, an evidence that association with, for those who use cannabis, individuals who have schizophrenia, and they're using cannabis, that these individuals do better cognitively than individuals who do not use cannabis. So that makes things complicated, right? Because it suggests that the use of cannabis, the association that we see could be self-medication. I don't happen to think that this is so simple. I think that probably there is a component of self-medication that also has a negative side effect. That is, you start off by self-medicating when you're, a teenage, maybe for anxiety, because you're prone to develop your developmental trajectory is such that you're prone to develop schizophrenia. You have the early stages, cognitive dysfunction, anxiety, and such. You use cannabis to get better, but that can actually aggravate your condition. Now, if you go online, you'll find all sort of statements about depression, suicide, and social anxiety. I'll tell you right away that these are very weak associations. They're either moderate association with small increased risk, and all are associations. So there is in the case of depression, suicide, and social anxiety, there is no evidence that this association has any causality involved. I want to point it out because I'm not a believer. I don't believe that cannabis is devoid of risk. I think that there are distinctive risks, especially in using cannabis in certain developmental stages. But I believe that we need to understand what those risks are. And sometimes we need to think a little bit outside of the box to identify those risks so that we are not barking at the wrong tree, if I could use that metaphor. But I want to go back to the NASM report quickly to, instead of just now looking at the risks, to look at the opportunities. So what are the medicinal potential, the medicinal potential for cannabis? So let me tell you two conclusions that the report made. Conclusion 4.0, actually, sorry, this is wrong. There is conclusive evidence that cannabis is an effective treatment for nausea in adults. There is also substantial evidence that cannabis is an effective treatment for pain in adults. So now the use of the adjectives conclusive and substantial is very determined. These are not picked out of thin air. Conclusive means certain things. Those of you who are involved in systematic reviews know what that means, and substantial means certain other things. So substantial is not conclusive, and conclusive requires more work. So if there is substantial evidence that cannabis is an effective treatment for pain in adults, we need to ask the question, as a society with limited resources, all societies have limited resources, if we want to move the needle from substantial to conclusive, we need an investment that could be pretty large. Is it worthwhile? So to me, the answer is yes, and is so yes for three reasons. The first is because, after all, we already have some evidence that it may work. And if it can work for pain, well, that pain is a big problem. Pain is a big problem, not only because of the pain itself, but because sometimes people, to fix the pain problem, use drugs like opiates that create their own problems. Like the opioid epidemic is not primarily due to people wanting to try the opioids on their own will, but because they got hooked on the opioids because of some pain-related matter, and they were prescribed opioids. So it seems to me that it's worthwhile because there is already some evidence. The second reason why I think it's worthwhile is that the health risk posed by cannabinoids is there, but is lower than those posed by the opioids. And we have to make the comparison not between the ideal world where we have an ideal drug that works perfectly for pain, because if we had that, we wouldn't be having this conversation today at all, but we don't. But what we need to compare the cannabinoids to is to the opioids. And as you know, opioids have, and as I said before, have multiple issues, including that of creating addiction. And finally, and that's where I spend most of my time, there is strong biological plausibility for cannabinoid regulation of pain. So it's not just something that we're pulling out of our hat. Oh, okay, so it maybe works. We have a huge amount of data suggesting that the endocannabinoid system plays an important role in pain. Before we go there, that's the rest of my presentation. I want to tackle briefly one of the one of the risks associated with the cannabis, and that's dependence. So we know that frequent use of cannabis results in dependence, and also in loss of control over use, which something we'll call craving, but actually is not the same as the type of craving you get with psychostimulants, in my opinion, completely different from opioids and psychostimulants, but can result in dependence. And that's obviously an issue. We know that sudden cessation of heavy and prolonged cannabis use can cause withdrawal. If one uses cannabis, you wake up in the morning, you use cannabis again at lunchtime, you use cannabis again at night, and by the end of your day, you have smoked maybe 4 or 5 joints, 6 joints. That is what I would call heavy use. If you do that for a period of several months, that's heavy prolonged use. And there is no question that if you stop cold turkey, you have withdrawal. Now, this is something that goes under in MSD5 in the statistical and whatever manual. It goes under the name cannabis use disorder, just like opioid use disorder. All right. Now, cannabis withdrawal is a serious problem, but I want to right away point out that if we think in terms of harm reduction, it's still better than the opioids, because opioid withdrawal kills you. It can kill you. The autonomic response is massive. There is an autonomic response with cannabis withdrawal, but it's not even close to that of the opioids. The symptoms are clearly disturbing and annoying, including irritability, loss of sleep, loss of appetite, loss of weight. These are all things that clearly are not nice and cause a lot of people to go back to use. But experiencing death again is not something that I know ever happened with cannabis withdrawal. So cannabis can cause acute toxicity. Everything does, even water, but there is no evidence really of fatality. And the main concern to me is not so much the acute toxicity, but the chronic toxicity after use in specific developmental stages of our lives. I like to focus on pain now because that's where the area that I remember was highlighted by the National Academy Committee as being an area of potential interest. And I would like to remind all of you how the pain circuit works. So the gentleman here hurt himself, and so the pain signal starts in the periphery of the body. These are C fibers or A delta fibers whose cell bodies are located in the dorsal root ganglia, and they go to the dorsal root. This is a long axon here. They go to the dorsal root, and from the dorsal root, they decussate. They cross over the other side, and they travel up into the brain stem, into the thalamus, and then eventually into the cortex, where we feel the pain response. So if we go and look at all these different relay stations, and again here we have the spinal cord with the A delta pain fibers, the C pain fibers arriving, sending second order neurons to the other side, to the contralateral side, and up to the thalamus and to some other sensory cortex 1 and some other sensory cortex 2, the amygdala, etc. All these different relay stations express cannabinoid receptors. And in most of those cases, not all of those cases, but in most of those cases, activation of those receptors inhibits the passage of the painful stimuli. Now, we have the receptor just like we have the opioid receptors pretty much in the same areas, not the same neurons, but there is not perfect cellular overlap, but there is regional overlap, structural overlap. The question is, do these receptors control pain? And the answer is a resounding yes. I'd like to give you one example first. We know there is such a thing as stress-induced analgesia. We all have experienced that. I don't know, those of you who play soccer or football or whatever game you play, or you like hiking or whatever, any physical activity, if you get hurt at the moment you are exerting that activity, you're focused on something, you want to achieve something, but you're stressed by that, you don't feel pain, the pain emerges afterwards. This is called stress-induced analgesia, and for years has been known to be mediated in part by the opioid system. And some time ago, we discovered with the lab of Andrea Homan that whatever is not mediated by the opioids is mediated by the cannabinoids. So this is one example, and there are many others, of a physiological function exerted by the endogenous cannabinoid system in regulating a very important physiological component of our response to pain. Interestingly, opioids and cannabinoids have similar effects, but they're not overlapping. Those effects are not overlapping, they're converging. And so one could also think that maybe we could leverage that to develop better medications that act on both, but that's a separate issue. So again, let's go back to the question, do cannabinoid receptors control pain? I'm going to do a little of archaeology with you today. And I found, this was last time I checked, last year, I found almost 3,500 papers on cannabinoid and pain published from 72 to 2020. And so let me go in archaeology. This is this paper we published many years ago, and we showed that peripheral endocannabinoids control pain. What you see here is simply a dose response of injection of anandamide in the paw of a mouse, which has also received an injection of formalin. Formalin is a very algogenic compound, has a proalgesic effect, so it causes a very massive pain behavior. Injection of anandamide into the paw along with formalin causes a very nice inhibition of that. If you inject anandamide in IV or IP, you have no such effect. And the reason for that is because actually we have cannabinoid receptors on C fibers as well as on A delta fibers, mostly on A delta fibers. And these are not either C or A delta, they're probably C fibers, but they're in the lung. And what you see here is work of Istvan Katona collaborating with us, showing the localization. This is immunogold staining, localization of Cb1 receptor in a C fiber innervating the lung. And something similar has been shown by Andrea Homan in C fibers innervating the skin, for example. So there are cannabinoid receptors on pain-sensing fibers that can be activated by endogenous cannabinoid in the periphery of the body. In fact, if you genetically ablate those receptors, so you remove them from the DRG, where the cell bodies for these fibers are located, when you do that, you enhance pain sensitivity and you block the pain-killing effects of THC. So these are indication of the importance of these peripheral Cb1 receptors and presumably the peripheral endocannabinoid signals in pain. In fact, we proposed a few years ago that there is a peripheral gate for pain, and that peripheral gate is localized at the very source of the pain signal. So this would be the ganglia, the dorsal root ganglia. This would be a schema of the spinal cord, and this is the terminal fields here. And all these different cell types that you see listed here release proalgesic mediators and analgesic mediators. We know that, and the analgesic mediators and the algebraic summation between these two opposing stimuli ends up determining whether receptor potential is produced. And if you produce a receptor potential, if you reach a certain threshold, you get to the action potential and you feel the pain. So the algebraic summation of the stimuli determines the generation of a pain signal, and we call that a peripheral gate. And anandamide, we have shown, and other lipids are important components of this gate. So back to the endocannabinoids, what I wanted to highlight here was that in this pathway, one can think of a lot of interesting targets. When you give THC or you use cannabis, what you're doing are basically activating an indiscriminately cannabinoid receptors, Cb1 and Cb2 throughout the body. You have no control. And the effects are could be potentially useful, maybe in pain. I mentioned that, of course, but you also have effects that you don't want because, but just by definition, you're activating indiscriminately all these receptors. So the line of thought over the last about 25 years has been, well, maybe we can optimize, we can interfere with endocannabinoid signaling in such a way that we maximize the benefits and minimize the risk. And so this is the idea behind the invention of fatty acid amino hydrolase inhibitors, Phi inhibitors. Phi is the enzyme that destroys an anandamide. And the idea here is that you destroy, by blocking this enzyme, you accumulate an anandamide. And because an anandamide is produced on demand, you only accumulate it where it is produced, when it is produced. So you have a more selective effect. So that's the basic for Phi inhibitors. And similarly, MGL inhibitors have shown, follow the same logic. You block them, you accumulate 2-AG, and presumably you enhance the effects of 2-AG without having the negative side effects of concomitantly activating all cannabinoid receptors that are not relevant. And so in addition, so the deactivating systems is what I just mentioned to you, and it's potential therapeutic targets. But of course, there are also, one could think about selective activation of Cb1 receptors, for example, in the periphery of the body, selective activation of Cb2 receptors, which are not psychoactive. And these are all have been investigated. If I went over all this, it could take me several days to go over them, but just remember that all these different targets have been explored. My lab has worked on Phi inhibitors. We generated the very first Phi inhibitor many years ago, the very first MGL inhibitor many years ago, the first inhibitor on anandamide transport many years ago. But I'd like to talk to you mostly about Phi inhibitors today. And in particular, I want to talk to you about one class of Phi inhibitors that I thought was of some interest to you. And these are peripherally restricted Phi inhibitors. So this what you see here is the scaffold of the first class of Phi inhibitors that turns out to be systemically active and selective. We discovered that back in 2003, almost 18 years ago. And you can see here the structure. This is a carbamate, and it's decorated. This is biphenyl group with this carboxyamide moiety that is quite important for activity. And what you see are here in our first Phi inhibitor, URB 597, which you see here. R is an H group, is an hydrogen group. And this compound has a very nice, it inhibits Phi throughout the body. It inhibits Phi in the brain very effectively. And in fact, it was even moved to clinical development. But then, unfortunately, the quandaries of science, of medical discovery are really particular. So that compound never really made it to the clinic, but it was moved toward clinical development, actually into clinical development. But if you replace now these R from hydrogen, you put in an hydroxyl group there, what happens is really interesting. The compound does not enter the brain anymore. And that's not because it's more hydrophilic. The PSA, the polar surface area is a little bit higher, but it's not that the reason. The main reason is because it gets pumped out of the brain by a couple of transporters, ATP binding cassette transporters, and that is due to this moiety and this moiety together. So that is the reason why URB 937 this is peripheralized, entirely peripheralized. And these are the two transporters. ABC means ATP binding cassette, and ABCG2 and ABCB1 are famous transporter. They're involved in the transport of a bunch of stuff. And interestingly, 937 is extruded from the CNS, but also from testis and fetus. So it's an interesting molecule and is now actually undergoing clinical development. So people have tested our compound in a bunch of models. I'm very proud that over 550 papers have been published on URB 597. Not as many have been published on, not just by us, but by many others. 937 has not been published as much, but maybe 30 papers in total. But these are models of pain where it has been found to be highly effective. And as you probably know, these are all mechanistically different models, but they're all peripheral models. They're models where there is a strong peripheral component, all right? So I'd like to just show you some data, and these are still older, fairly old data, showing the effectiveness of phi inhibition in our own work. So this is a model called the mouse chronic constriction injury model, and it's a model of neurogenic pain. So it's not neuropathic, it's neurogenic. The pain starts in the sciatic nerve, so in the periphery, it's accompanied by inflammation. But one of the signs is so-called mechanical allodynia. For those of you who are not into this line of research, allodynia is that phenomenon whereby one feels a non-painful stimulus as being painful. For example, you go to Tahiti one beautiful day and you go to the beach and you love it. It's such a wonderful place. So you start snorkeling and you snorkel for 4 hours. You forget about the sun. You go back home and you realize that your back is basically burned to no recognition, and from then on, for the rest of your vacation, every time you take a shower, you die because it's extremely painful. So something that is not painful, a shower, becomes painful due to an injury. So mechanical allodynia, though, is basically poking an area around the constriction injury, and you can see this. So you have a withdrawal threshold in grams. So normally the animal withstands about 4.2 grams, but after being exposed to this chronic constriction injury, it goes from 4.2 to about 1.8. So there is a substantial dynamic range here. And if you use this beautiful drug here, gabapentin, it completely blocks this effect. It's 50 milligram per kilogram by oral dose gabapentin, which I can tell you is a great drug because I'm actually taking it right now because of back pain, and Chris knows that I've been limping around the building recently. Is a good drug, but has a lot of side effects. For example, I didn't take it this morning because if I took it this morning, I would be really having a hard time collecting my thoughts and talking to you coherently. So that it's a good drug, but not a perfect drug. So here is the effect of our peripheral phi inhibitor. And so this is something that does not make it to the brain. So there is no way this thing can cause sedation or mental confusion. And these are the dosages. They are substantially less than gabapentin. And you can see that the 3 milligram per kilogram oral doses, it completely abolishes the pain response. Let's go to another model. This is the complete Freund adjuvant model. Is a model of inflammatory pain. So dexamethasone is a nice, very powerful steroid, not the most powerful, but fairly powerful, and has a nice effect on allodynia. However, if you look at 937, which is here, 937 at a maximal dose of 30 milligram per kilogram orally has a much greater efficacy than dexamethasone, and you have a very nice dose-dependent effect. I'd like just to point out that this 597 is our own centrally active phi inhibitor, and PF stands for PF7845, which is a Pfizer compound, a Pfizer phi inhibitor, which was brought to the clinic. And you can see either they're chemically different. You can see that either our own compound of the PF compound, which we synthesized and test because the company wouldn't give it to us, they don't achieve maximal efficacy. The maximal efficacy is produced by peripheral inhibition of phi, excluding a central inhibition. That's a topic for some interesting research that needs still to be done. Now, in closing this, I'm at 50 minutes. I'd like to leave some time for questions and discussion. I'd like to mention some evidence from human research that suggests that phi could actually be important for pain. So there are spontaneous human mutations in the phi gene. And there is one famous called C385A, which is hypofunctional. This hypofunctional mutation causes the enzyme to degrade more quickly. So there is less phi and there is more anandamide in the body and in the brain in particular. And these individuals tend to have less pain. They also have less anxiety. But there is one individual so far that has an even more radical mutation, and that radical mutation is called phi out, and that was recently discovered as follows. This lovely lady here, who is in her 70s now, at the time in her late 60s, had to undergo surgery. So she went to her doctor, and the doctor prescribed to her some painkillers, to which she said, well, thank you so much, doctor, but I don't need them. the doctor looked at her and said, "What? You don't need the painkillers? You know, you're going, knee surgery?" I said, "No, I mean, I never really take painkillers, and I have two boys, and I mean, two young men at the point, actually, two grown-up men. But when I gave birth to them, I didn't need any painkiller." So that kind of, this was in Scotland, and so the practicing physician who was talking to her had the intuition of contacting the UCL, the University College London, where they are expert at genetic analysis of pain, and they genotyped this lady and found that she carries two mutations: the C385A, which is the one I mentioned to you, plus this far out. And the far out mutation, the C385A mutation together, basically remove FAR from her body. So there is no FAR, she has no FAR, and pretty high levels of anandamide and other FAR substrates. Interestingly, her father also had the same mutation, very likely, because he also needed no medications, but her son had only C385A, and he needed some pain medication. So although these are very, of course, very very limited studies, they're not a phase 3 placebo-controlled studies. They give us hope and promise that in the future there might be medications that target FAR to relieve pain. One thing that I like to say, and I also mention to my medical students, is usually that when I teach them pain, is that unlike other innate analgesic conditions, this is not associated with early death, because usually people die because they hurt themselves. She hurts herself a lot because she doesn't feel the pain, but she also heals very quickly. And that is also due to FAR, but it's the lack of FAR. But that's another story for another paper, for another talk. I will stop here. Thank you for your attention, and take any questions. We have still a few minutes.
Thank you very much. We have already a few questions. Let me ask a very intriguing proposition here that you made. If we would have a ligand that is not penetrating the central nervous system and only, will address the inhibition of fat tissue, would that be a good drug that will lower the weight?
Yes, absolutely. In fact, there are companies, a few biopharmaceutical startup companies that are looking into that. And there is very promising preclinical data that a peripheralized Cb1 receptor antagonist will have very profound anti-obesity effects without the psychological side effects. The reason for that is because there are cannabinoid Cb1 receptors in the adipose organ, both in white adipose tissue and brown adipose tissue, and they inevitably have the same overarching logic. They stimulate lipogenesis, so and as well as the creation of new adipocytes. So they basically favor the accumulation of fat. So the blockade of those receptors is indeed does indeed reduce body weight, not by working on food intake, but by working on by blocking lipogenesis and stimulating lipolysis.
Mm-hmm. Yeah, so there is a hope on that. And my final question before we go again to Greg and Zach, do you know what role of those compounds playing for physiology of the plant? And
it's an interesting question. I love that question because it's a question I always ask myself, for cocaine, for nicotine, etc. For some stuff like nicotine is obvious. Nicotine actually kills bugs. It's a very powerful poison. For THC is a little bit different. So one thing you need to know is that the plant does not actually produce THC. The plant produces a precursor for THC called THC COOH or THCAA. And that compound has no affinity for cannabinoid receptors, a little bit for CB1, but very little. But in order for us to enjoy cannabis, the THC, the carboxyl group must go. So that decarboxylation happens with heating the plant or drying the plant. So it's THC-COH what the plant makes. And what people think, there are a few papers out suggesting that actually what it does, it causes apoptosis in specific cells of the cannabis plant when those cells are not needed. So it looks like, again, some kind of signaling mechanism in the plant, but not an insecticide mechanism, which is not possible, because one think, okay, the animal eats the plant and gets, go crazy or goes, no, that can't happen, because THCOH does not get even to the brain.
Okay.
THCOH doesn't get to the brain.
Thank you so much. Let's move to Greg.
Could you comment a little bit on the chemical diversity of endocannabinoids like arachidonyl taurine and arachidonyl dopamine? And the one that I would be particularly interested in is arachidonyl taurine, specifically because in the eye, it has extraordinarily high concentrations of taurine in the eye, much more than most tissues within the body.
Well, that's a wonderful question, and the topic for a presentation I could give today, because we've worked a lot on taurines, although we only published one paper on them. So for those who don't know, N-acetyltaurines are a class of molecules that are also substrates for Pha. Most of them do not activate, none of them activates cannabinoid receptors, including N-arachidonyltaurine. And the reason for that is because taurine is charge, is a negative charge, actually two negative charges at physiological pH, so it can't get into the receptor site. We don't know what the receptor of the taurines are, but they're not considered normally endocannabinoids. Likewise, the N-arachidonyl glycines are not considered endocannabinoids. Some erroneously, people sometimes call them endocannabinoids. A lot of us scientists who are so strict when it comes to research and experiments and data and control tend to be a little bit lackadaisical when it comes to words. And I am not that way. I'm probably even more of a stickler with words than I am with other stuff. I really think that we should not use the word endocannabinoids for the taurines, for the glycines, and the dopamines. Now, N-arachidonyl dopamine may have some effects on Cb1, but I doubt it. They are interesting molecules, and their physiological roles are unknown. Now, the N-arachidonyltaurine in particular, I did not know that was highly expressed in the eye. It's quite interesting. We looked at long-chain saturated taurines in the skin and found that they are extremely important in regulating skin wound healing. In fact, they are pro-healing or pro-reparative molecules, without which there is a substantial problem in healing. And when you enhance their levels, the healing is accelerated. And actually, that is the reason why I think that lovely lady you saw could hurt herself. She injured herself, but in fact, in the paper that I cited there is a lovely photograph showing her arms full of injuries, all perfectly healed, with a very tiny little scar. And we have shown in animals that Pha knockout animals healed perfectly, a lot faster than wild-type animals do. So it is possible that N-arachidonyltaurine in the eye, which of course has got a lot of epithelial tissue and all sort of interesting things, which you can teach me about because I know nothing about it, but it is possible that it has also a role in healing. It would be really important to understand the receptor for these taurines. As I said, we do not know what the receptor is, but it's certainly not Cb1.
Okay. Just to be clear, I don't know about N-arachidonyl taurine, but just taurine itself is in super high concentrations. But
taurine itself?
Yeah, taurine itself is in extremely high concentrations in the eye.
Well, that is really, really important to know, because actually taurine is essential for the synthesis of N-acetyltaurines, because the synthesis of N-acetyltaurines is dictated by the taurine transporter. The taurine transporter that transports taurine from outside inside the cell, if you don't have it, there are no N-acetyltaurines produced. So taurine is limiting to the rate of N-acetyltaurine biosynthesis. And I suspect, based on what you just told me, and what we know about taurine and healing, that these molecules could be really important in healing of the eye. And in fact, what I would recommend, what I would suggest is, as we talk more about this, we have a Pha knockout that selectively does not recognize N-acetyltaurine. So that's a Pha knockout in which Pha cleaves everything except the N-acetyltaurins. And it's a model that could be used to specifically ask the question if there is a physiological role of N-acetyltaurins in eye physiology.
Thank you very much.
Zach?
Hi, that was a really cool talk. I had a question about glial cells. So do you know if glial cells like in the central nervous system or the eye co-express Cb1 and Cb2 receptors? And if so, is there like a specific ratio that could, of like Cb1 versus Cb2 stimulation that could bestow like the best neuroprotective effect of those cells to build like and actually enhance their neuroprotective?
Right. So that's a lovely question. Thank you so much, Zach, for asking it. So CB1 astroglia have low levels of CB1, but there are people out there who think those, even though very low, those receptors are really important. I don't know for a fact, the number of papers is limited, but there are high profile papers showing that if you knock out CB1 receptor in astroglia, all sort of stuff happens. Well, I suspend my judgment 100%, but I do see that's a possibility. As far as microglia is concerned now, so microglia, quiescent microglia probably expresses CB2, but not in great amounts. But you clearly, when you stimulate microglia, say with LPS and the toxin, you have CB2 expression. And most importantly, in my opinion, monocytes, circulating monocytes, travel when you have an inflammation in the central nervous system, or you have signals that are neuroinflammatory in nervous system. The monocytes travel to that area and they carry their Cb2 receptor with them. So even though the astroglia and microglia may not have huge amount of it, they could still be coming from the periphery, and that those receptors are active, and we know that they are active.
So is there like a specific, is there like a specific ratio of CB1 versus CB2 activation?
I don't know about that, but the CB2 typically is in minute amounts, much lower than in any other, much lower than in any other. Oh, it's all black now, and for some reason I must have clicked something wrong.
Okay, thank you.
Ben Lin.
Hi, good talk. So I'm curious about the dependency, and so is there any way to isolate to make it less addition and them, but to keep the Q-pillar effect?
I'm not sure I understand 100% your question. You're asking about tolerance, maybe, or depend?
No, the dependence.
So your question is whether one could use the effect on the dilating effect on the eye. Is that your question? Without causing dependence?
Yeah, but not just for the eye, for the pain. So to control the pain, and yeah, that's most important, especially for a lot of patients. And so, but in the meantime, especially for some regular people without any pain, or just like, it's also related to another question about the policy. So do you think should it be a controlled substance, or it should be legal for anyone to get it or something?
Okay, so I can answer the first question more intelligently. So basically, it is impossible to separate completely THC-induced tolerance and dependence from THC-induced pharmacology, and the analgesic effect is clearly linked also to dependence. But as often happens in this case, tolerance can be managed, and can be managed by using lower doses, which are typically sufficient to, in fact, are required to produce analgesia, because if you increase the doses, the analgesic effect disappears. In most clinical trials, the effect is obtained at low doses of THC in cannabis, not at high doses. At high doses, you actually start having the opposite effect. So to me, so let me just frame this in a general concept. To me, cannabis is not a good medicine because it's a difficult to use medicine. And I like medicines that are easy to use. I don't want to be worrying about, taking a drug that, if I take twice instead of taking once makes me high. I don't want that. So my basic thought process that cannabis is not a good medicine, but it is still good enough and probably better than opioids in a number of different ways. So if one is able to use cannabis appropriately for analgesia, one, I think, can achieve a substantial effect. I think also without tolerance, if you use appropriate dosing. Diversion is always a problem because you give someone, something and they can always misuse it. But I think the misuse of opioids has a great amount of risk and carries a lot of problems. The misuse of cannabinoids is much less risky. So again, we're looking at a harm reduction and balancing act here. And there is, I think, a place for cannabis in that way. Now, for the eye, I don't know, I haven't heard the question yet, but I assume you guys are interested. You know what the American Ophthalmological Association rather has said. Indeed, cannabis does cause a decrease in eye pressure that has been known for years. People have used for glaucoma. However, this effect is short-lasting. And I think the American Ophthalmological Association took a very sort of conservative stand on that because they said, well, this, the effect is too short-lasting to warrant the risk. And the risk being what? The risk, that they highlighted was dependence, which I don't think honestly is a risk if you apply the compound locally. I think actually probably the risk is zero in the case of dependence. But they also were concerned about vasodilation, because then they said vasodilation can cause decreased irroration, vascular blood supply to the optic nerve, and that could be problematic. That I also think is not a valid criticism, because I think if one uses an appropriate dose of THC in a collyrium form, one can obtain very substantial effects without, I think without, and it has to be, of course, appropriately formulated, and people are working on this, and it has to be slow release, and people are working on this. But, I think taking a stance that so universally banishes cannabis because of this purported side effects, I think is irresponsible, because it eliminates a therapeutic opportunity for people who really need it, as you guys know.
So let's have a last two questions, and then maybe with final comments, we'll go to Peter Cullen. So let's start with Alex, and then Magdi, and then Peter, all yours.
Thank you, Chris. Thank you very much, Dr. Piomelli. Beautiful talk. I have a quick question, if you can hear me okay. There's some evidence of Cb1 being expressed in high levels in enteric nervous system, in submucosal cells and the interstitial cells of Cajal. Do you think that it plays an important role in, say, the brain-gut axis, this cannabinoid signaling, or in something to do with enteric nervous system signaling?
Lovely question. Thank you so much, Alexander. Yes, indeed, there is a clear role. And so there are cannabinoid receptors and endocannabinoids throughout the gut. In fact, we find them in the lower and the upper and lower intestine, where they serve a multiplicity of functions. They're present in the enterocytes, they're present in the macrophages and local macrophages, they're present on the fibers, and they're present in the plexi that form the enteric nervous system when they control the release of transmitters. So depending on where they are, they have different functions. In the enteric nervous system proper, the one function that I know about is the release of acetylcholine and the propulsion, the ability to control propulsion. And in animals, at least, cannabinoid activation has a slowing effect on bowel movements. In humans, this is not a very prominent effect, as anyone who has ever tried cannabis knows. Unlike oxycodone and Vicodin and all these opioids, cannabis does not cause constipation. But in animals, there is a role there. So there are Cb1 receptors and Cb2 receptors in the gut. One aspect that is interesting, you asked about the gut-brain axis, is that Cb1 receptors in cells, in select cells, of the gut. My former postdoc and now faculty at UC Riverside, Nick DiPatrizio, showed that are involved in the regulation of fat appetite. So when we eat fatty foods, we typically have we have a specific appetite for fat. We feel fat. Fat is one of the tastes along with the other known tastes. When we feel this, cannabinoid receptors are endocannabinoids are produced in the upper intestine, in particular in the jejunum. So there is a flood of cannabinoids that are produced there. They activate cannabinoid receptors, and that through a mechanism that probably involves the vagus tells the brain continue eating. So you feel fat in your mouth, the signal goes down to the intestine, goes back to the brain, and the brain, now regulating behavior, says continue eating. And the reason for that, I believe, is because, I don't know, etaologically, that makes sense, is because fat is so rare in nature, and whenever you find some fat, you need to eat as much as possible. When you're done. But as long as you have available fat, it's good to eat it. Anyone of you who is interested in anthropology knows that our ancestors would cut broken bones and suck out the marrow, because that's very fat and very nourishing. So that's an interesting role for cannabinoid receptors in this gut-brain axis control of fat eating. I also have a question for you, though, Alexander. I see that your background is at Greek temple. Is that a Greek temple in Paestum?
No, no, it's not. It's something my kids had built, and through serendipity, ended up looking like something important. But I assure you, it's nothing of that sort.
All right. Well, it looks really good. I thought it was a Greek temple.
Maybe there's a future for that.
Yeah.
Magdi.
Thank you. Thank you for the lecture. I have two questions. Are Cb1 and Cb2 receptors found in astrocytes? And generally, do you think astrocytes involved in this healing process that you mentioned, or the dependence process of the cannabinoids?
So yes, there are Cb1 receptors in astrocytes in fairly low numbers, and if you remove them, things happen. There are a couple of papers out there, very prominent papers. I think the literature is not strong enough for me to make a strong statement, but clearly Cb1 receptors are important in astrocyte function. Are they involved in what we recognized as the main effects of cannabinoids? We don't know. But probably not. Probably the most important cannabinoid receptors involved in intoxication, in the memory deficit effects, in the rewarding effects, et cetera, those are probably Cb1 receptors in neurons. Now, outside the brain, every other single cell of the body has cannabinoid receptors, including adipocytes, enterocytes, and hepatocytes, and myocytes. They all have Cb1 receptors in low numbers, and there is pretty strong evidence that in each of those cells, cannabinoid receptors and endocannabinoids play specific modulatory functions that can be very important, as in the case of the adipocytes, or less important, as in the case of the myocytes. But in all cases, there is an overarching logic, and is regulation of energy balance, because that's what endocannabinoids do a lot outside the brain. They regulate energy balance.
Peter,
Thank you.
Ready to share your final thoughts? Well, if not, then let's go to Philip Kaiser.
Hi, Dr. Piomelli. Sorry, I'm sorry, I messed your name up. It's fantastic talk. I guess what I was kind of wondering is, and I don't follow this field very closely, but kind of going back to the pharmacognosy of the cannabis plant, are people doing any genetic engineering on this to try to block specific, metabolic pathways, or prevent the compound from going to completion to the precursor that you described, to see like what effects that has? I was just curious with the, if research on that has kind of picked up now that the genetic tools are really widely available with CRISPR-Cas9 and so on.
Yeah, no, there is. Thank you. There are a lot of course, this is not my area of expertise. So what I'm going to tell you is what I read, right? So, but there's been now for the last 5 years a huge amount of interest in this, because the legalization of cannabis has brought about an opportunity, but also a problem. How do you use it, right? How do you actually make money with it? And the best way to do that is to generate clones that are unique to you, then you can sell to people, and their clones are uniquely useful at doing something. So one thing about cannabis that is interesting is that it's really a chemical factory. So the 140 cannabinoids, 141, 42, the number grows every day. Those compounds, they have a common precursor, and particularly because they come through the classical pathway of the terpene biosynthesis, right? So they're terpenophenolic compounds. So the precursor for example, the precursor for THC and CBD is common, right? There is a common precursor, and then there are enzymes that are specific and it lead either to THC or to CBD. So you can actually very simply make a plant that only produces CBD by removing the THC, the enzyme that leads to THC. That's pretty clear. But likewise, you can tweak the chemistry in similar ways by changing other enzymes and forming other cannabinoids like cannabichromine and cannabigerol and cannabidivarin and cannabidivarin, all these various compounds whose pharmacology is really not well understood. So there is a lot of interest now in pharmaceutical companies. Okay, the plant is available. We can mess with it genetically. We can make a plant that only makes one particular metabolite. But why would we do that? Is that useful? So they're looking into these different pharmacologists, trying to understand if there is money to be made, if there is value to be developed. Another interesting thing is that you can actually remove all the enzyme from the plant. You can just transgene them into yeast, and you can produce THC in yeast, and you can produce CBD in yeast. And whether that is commercially interesting, I really don't know. But it seems it is interesting from a legal perspective, because whereas the cannabis plant is illegal, yeast making THC would not be illegal. So, a lot of people are thinking, oh, maybe we shouldn't use this pathway because the federal government is never going to change their mind. I actually think that the federal government will change their mind. I actually think that we are very close to that. Not that they speak to me, but I think it makes complete sense. Is one of those things that would make complete sense in the next couple of years. And I would not be at all surprised if we had a major sea change in the legal landscape on cannabis with the Biden administration. But maybe it's my optimism. But back to your question, yes, the answer to that is yes. There are a ton of people looking at that, entire companies dedicated to that.
Thank you. Sorry again for butchering your name.
That's okay. I'm used to it. I mean, I put myself in Chris's shoes, and I think, which are Gucci, I understand. I in Chris's shoes, and I think, oh, poor guy, his name must be butchered even more often than mine. So I'm happy with that. I don't mind.
All right, thank you very much. We had a wonderful presentation and great discussion.
Thank you so much for having me, Chris. And you know where to find me. So if you need me, and I'd love to talk if you guys think that this taurine story is interesting. I'd love to talk about that.